and one time the cell was divided. Meanwhile, in meiosis, we have also one time the DNA was duplicated. And two times the cells was divided. And as a consequences from this, in mitosis, we have from one mother cell that produce two daughter cells, which have the genome, genetic background identical to the parent cell. Meanwhile, in meiosis, we have from one mother cell produce four daughter cells. Okay, and each daughter cell we have the genomic decrease by a half compared to the parent cells. And not only the number of chromosomes was decreased by a half, but the genetic background of the daughter cells is a little bit different due to the recombinant because of the crossing over during meiosis 1. In the metaphase 1, 2 double chromosome line up in the metaphase plate on the equator of the cell. So there's crossing over between two regions of the chromosome, and it's produce a new recombinant chromosome with part of chromosomes is belong to the homologous chromosome. And so the mitosis is the way of cell division in the somatic cell, and also in the primitive sex cell, that the immature sex cells. And meanwhile, the meiosis is the way of cell division in the mature sex cells. Okay. And so in last week, we have also discussed on the, a minute. And for the rest, could you unmute yourself? Okay, I'm unmute you. So in last week, we have also discussed on what is the differences first on the transcription and translation of the prokaryotic cell and the eukaryotic cell. Okay. So the first question is in prokaryotic cell, because we don't have the nucleus membrane to separate the nucleus region to the cytoplasm. So we will have the transcription and translation was happen continuously. So as soon as the RNA polymerase or transcription factor come and start to synthesize the mRNA, the ribosome will come and binding immediately into the mRNA and start to synthesize the polypeptide. Okay. Meanwhile, in the eukaryotic cell, because of the nucleus membrane here separate the nucleus cell with the cytoplasm. So we have the transcription happen inside the nucleus membrane, and then the mature mRNA will having been through several processes that we discuss later to cut down the intron and binding the 5' cap and poly A tail to be to develop into mature mRNA, and the mRNA will get the outside of nucleus membrane through the nucleus hole on the nucleus membrane, and then the translation start after the mRNA had been in the cytoplasm. The ribosome come and binding and start to polypeptide. So this process is happen discontinuously. Okay, with the transcription happen inside the nucleus region, and the translation happen inside the cytoplasm. And one more thing is the size of the ribosome in the prokaryotic cell is a little bit smaller, which is 70 S, with the S is a unit of centrifuge fork. Follow the scientist name of the scientist who invent this unit, and consists of two subunits. The small one, which is 30S, and the last one, which is 50S. And meanwhile, the ribosome in the eukaryotic cell is a little bit larger, with 80S, which combined of the small 40S and the last one with 60S. Oh, by the way, I remember that in this class, last week, there's a student who asking about why in RNA we have uracil instead of thymine in DNA, right? So I checked for the research on PubMed. So it's turned out that the T in DNA is more stable because it will quite resistant to the photogenesis mutation in the nucleus. So with the thymine in the DNA is more stable and maintain the function of DNA is to storage and duplicate the information. However, if the thymine is go to inside outside of the nucleus, so this is the nucleus. If the thymine is go outside of nucleus, so the thymine is quite stable inside the nucleus, but when it gets outside in the cytoplasm, it's quite susceptible to the oxidation in the cytoplasm. So as a result, the thymine will be easily to be destroyed outside of the nucleus region. So that's why the RNA must use a similar structure, uracil, which is more resistant to the oxidative in the cytoplasm. And of course, the uracil is not quite stable to the photon mutation, but it's quite stable to the oxidation. So the RNA, because the RNA have to move out of the cytoplasm, so uracil could be the better material. Okay. And in last week, we have discussed also on the promoter of the prokaryotic cell and the promoter of eukaryotic cell, and we discuss on the differences in the DNA replication and RNA transcription. So in here, DNA replication using both templates as a strand, both DNA strand as a template to synthesize the new DNA strand, follow the semi-conservative rule. And in transcription, it's used only one template, only one strand as a template. So the product of the DNA replication is double-strand DNA because it's using two DNA double strand as a template, and in here is a single strand RNA because only one strand was used as a template. So the substrate in here is deoxy nucleotide, and in here is ribose nucleotide. The primer DNA replication require the RNA primer. Meanwhile, the RNA transcription requires no primer. Okay, so the enzyme participates in processing here is DNA polymerase, and in here is RNA polymerase. So we discuss also on RNA transcription. So in here, it using only one DNA strand as a template, which is the sequence from 3' to 5' here. So the RNA polymerase will binding on only on this one, and that's why we call this one is a template strand. The other sequence here, the other strand here, will have the sequence the same with the mRNA. So replace only the T thymine by EU uracil. So we call it the DNA coding strand, because this strand have the same sequence with the mRNA here. And sometimes we call it the positive strand. Okay, and the other strand here is used as template. We call it the negative strand. Okay. Wait. So come to today's lecture. So first, let's discuss on how gene was structured in the prokaryotic cell. So in here, you can see that this is the promoter regions. Oh, promoter. And this is the position +1. +1 is also called TSS, which stands for transcription. Starting site. Transcription starting site. So the promoter will binding in here by recognize these two regions, is binding here, and slide until it reach the +1, the TSS, and it start to synthesize the mRNA here. So this is the same between the prokaryotic cell and the eukaryotic cell, but the difference is in here, several genes will organize together as a structure called operon. So operon is several genes which have a similar function that encode for quite a similar protein, or the protein with the same similar functions that share structure organized as a structure called operon, and they share the same promoter. Okay, so whenever the operon was activated, so the transcription factor binding in here, and it will transcribe the whole mRNA, which is encoding for different unit of protein. Okay, so genes here is defined as a unit for a unit of protein. So in here we have different genes organized as an operon. Let's watch a small clip to this demonstration on this process. So this is the nucleus region of the prokaryotic cell. So fragment. So this is the two typical region that the promoter, that the transcription factor recognize. This is the promoter is have binding in the -10 with the typical TAAT sequence, and the 30 -35 with the TTGACA sequences. Okay. Okay, so transcription factor binding in. Okay, so again, you can see that in here, the now strand the DNA strand in the below here is called the DNA template strand or the negative strand. Okay, so the sequence here is the non-template strand or the coding strand of DNA, or the plus one, the positive strand. Okay. Okay, so the promoter binding, the transfer factor binding in promoter, but it not start to synthesize the mRNA right there, but it slide until it reached the position +1 or the transcription starting site. Here is a transcription starting site. Okay, and it starts to synthesize the mRNA from this position. Okay, so the mRNA here, the mRNA was synthesized, follow the Chagas rule. So you can see that in prokaryotic cell, the transcription and translation continuously processed. So as soon as the transcription had not finished yet, the ribosome have come and binding into the mRNA to start the translation process. So this is a continuous process. Transcription and translation would happen at the same time. So as long as the transfer factor reach the termination region, it will release the mRNA and the transcription process finish in there. So come to eukaryotic cell is a little bit different. Where's my? So it's the same region like prokaryotic cell, that it including the promoter and the transcription factor when binding in here. Transcription factor. And it slide until it reached the position +1. So this is a transcription starting site. But different with the prokaryotic cell, that in eukaryotic cell, it had the region called the intron. So as long as the premature mRNA was synthesized, it's including all the region, but it had to been through the process called splicing. Splicing to remove all the intron region here and put the 5' cap and the poly-A tail in here. So the typical structure of the mRNA is in the slide you can see here, with the 5' cap, the 5' UTR, the coding sequence. Coding sequence in here is coding for protein. For protein. And 3' UTR. So maybe I will open painfly to explain more clear. So this is transcription and translation. So in here we have a double-strand DNA that encode for genes. Double-strand. Because it's encoded for gene is will have its promoter. So let's hypothesis this is a promoter region. So this is and as the promoter region, the transcription factor will binding in there here. Binding in here and start to synthesize. But it will not start to synthesize the mRNA from this position. It starts in here, in the position +1. This is position +1. Stand for TSS. So different with the gene in the prokaryotic cell. In prokaryotic cell, we have intron. That here is intron. We have an intron here. Okay. So the transcription factor binding in promoter region slide until it reaches the position +1 and it start to synthesize the mRNA here. So this is the premature on A mRNA with the sequence of intron. Okay. So this is translation. Sorry, it's transcription. And the premature mRNA will been through a process to become mature mRNA with the intron was removal or splicing, and the 5' cap putting here. This is 5' cap. And poly-A tail. So this poly-A tail, that means it's a, and thanks to this poly-A tail, whenever we want to catch the mRNA to investigate, for example, the transcription level of a specific gene, we will put in there the primer, including the poly-T, the oligopoly-T, to catch the poly-A tail. So in here, the intron was removal.
Excuse me, can you tell me again what does the poly-A tail do?
So the poly-A tail in here during, I don't know if I should share. So the poly-A tail here is the structure of poly-A. So it including many A, and it attacks into the 3' direction of the mRNA. Okay, and in order to catch this this mRNA to investigate, for example, a transcription level, the scientists will design the oligo T to have it binding with the poly-A tail to catch the mRNA. Okay, it's a structure formed during the process of the mRNA was mature. Okay. Right. So this poly-A tail, and the intron was removable. Remove. Splicing. And during splicing process, different exon will be removed too, but we will talk about it later. So this is the mature mRNA. So it's going to translation process. So that means that so the position +1 will be maintained in order here. +1. So as a consequence of the translation, the ribosome, so this is 2 subunit of ribosome, will come and binding in the mRNA and start to synthesize the polypeptide. But like the transcription factor in here is binding in promoter region, but it slides until it reaches the transcription starting site. So ribosome will binding in here, but it will not start to synthesize the protein immediately. It will slide until it reaches the start codon. For example, this is the start codon. Start codon is AUG, which is encoding for methionine. So from this position, it will start to synthesize the polypeptide. So the first peptide is methionine here, and we'll continue to start to synthesize another protein, another amino acid here. Until it reached the stop codon, right? So in universal coding sequence, we have the stop codon is UAA or UAG or UGA. Okay. So in this position, the polypeptide was stop synthesized and it released and going to is the post-translation modification. But in here, we have the regions from the position +1 here into this. We have the sequence, the region here from this point to this point. Okay. So the mRNA, which is the transcription starting site, was in this position, but the ribosome will not start to synthesize the polypeptide until it reaches the start codon. So this is the translation starting site, and this is the transcription starting site. And this region of mRNA was transcribed but not translated. So that's why we call it 5' UTR. Oh, maybe I will choose another color too. 5' UTR or 5' untranslated regions. Okay, so the position from this to this, and the same with the position here from the stop codon to the end. So we have the region here. This region was transcribed. It's located in the mRNA, but not translated because this thing is not coding. So we call it the 3' UTR regions. Okay. So like the structure of the mRNA here, we have the 5' UTR and the 3' UTR here. So this is the position of +1, the transcription starting site, and this is the start codon. All the region in here is 5' UTR. So some of you have participated in the bioinformatics practical training. Do you remember the exercise number 5 here? So in exercise number 5, they want us to explore a gene called MYH9, right? And in question 3, they want us to look at in the transcript table in the transcription of MYH9-201 and see which exon is completely and partially untranslated. So to explore that, we go to MYH9. So for the student who have not attended to the practical course, you go to Ensemble, which is a very large genome database. Click into human, explore the MYH9, click on go. You click on the human genes, and here we have observed different transcript variant of the MYH9. What is a transcript variant? We will discuss in a few minutes. But for the moment, just focus on the transcript MYH9-201. Okay, you can click in here. Oh, it don't provide a picture here. Maybe YouTube. Okay, so the MYH9-201 in here, it including, as I remember, 41 exon. Yeah, 41 exon. But among those, only 40 was coding exon. Okay, so we can see in here, in this figure, it including 41 exons. So the last particle, small but thick particle here is exon, and the small and thin line here is the intron. Okay, and if you observe an exon with a dark color in here, that means that it encoding for protein, and the blank exon here, the square with a white blank in the middle here, is the non-coding region. So we can see that all the exon in the middle was coding for protein, but we have a part in the beginning here is not coding for protein, and the part in the end of the gene here is not coding for protein. So because this is the 5' UTR of the genes, and this is the 3' UTR of the genes. So that means that this region, yeah.
So are there the big, there are the large vertical part, is it, are they the intron?
I'm sorry, what?
Are there the intron, like the brown thick part and vertical part?
Yeah, I just explained. So this small line here is the intron. So here is the intron. Here is the intron here. Okay, do you see my point stick?
Okay.
Yeah, so this is the intron, and this small here is the exon. Okay, exon, part here, exon. Okay, you see it? Okay, so all the fragment here is the exon. Okay. Okay for you to recognize. So in here we have 41 exons, so it's a very large gene. And the part of here is not coding for protein. That means that this is the 5' UTR, and this is the 3' UTR. Okay, and in the middle here is the CDS. That means the coding sequence. Okay, right. So one phenomenon is quite interesting also is the alternatively splicing in prokaryotic cell. So that means that not all the time, all the exons was transcribed into mRNA. So for example, in the promoter here, so this is transcription starting site. We have a gene with 4 exons, exon number 1, 2, 3, and 4. So in one way of splicing, the mRNA will be synthesized with the sequence from all the exons, exon number 1, exon number 2, 3, and 4 here. So it's a classic splicing. But in alternative way of splicing, where the process somehow overskip the exon number 2, so the new mRNA now have include only the exon number 1, number 3, and number 4. So this is called the alternative splicing, and it's called to the transcript variant that we have just observed. just observe in the MYH9 here, we have so many transcript variants. We have at least 6 variants is coding for protein, and the rest is not coding for protein. Okay. So how is so? So another example in here, we have a gene with 1, 2, 3, 4, 5, 6 exons. And the splicing was including, because of the different way in splicing, in the one type mRNA, we have including only exon number 1, 2, 4, 5, and 6. Somehow the exon number 3 was skipped in this type of translation. And as a result, the protein number 1, including only the region from 1, 2, 4, 5, and 6, but not 3. Another way is to the mRNA was splicing based on exon number 1. It's overskip. Exon number 2, 3, overskip. Therefore, 5 and 6. So as consequences, we have the protein including only 1, 3, 5, 6. And in the another way of splicing, we have only the 1, 3, 4, 5, 6, and the 2 was skipped. So like in here, we have different ways of splicing. So in among 6 transcript variant that encode for protein, the largest one is the 215 here. Okay. And is encoding for a protein, very big protein, 1981 amino acids. Meanwhile, the smallest one variant is coding for only 103 amino acids. So you can see in the table the relative size of the protein here. So the largest one is the 215 here, and the smallest one. Okay. Somehow it's overskip all the 1, 2, 3, 4, 5. So this one is including only 3 exons, 1, 2, 3, and this one is including nearly 41 exons. Okay. And the higher vertebrate, the higher level organisms, the more way of alternative splicing is might have. So scientists have found out the in Drosophila, the DS-CAM1 gene have over 38,000 transcript variants. So that means that this gene was encoding for more than 38,000 transcript mRNA. So this is research published on Genome Research, have found out that in the human genomes, in 25,000 genes, human genome is composed of 25,000 genes. Only about less than 5,000 genes was encoding only for one mRNA molecule. About nearly 3,000 is have 2 transcript variant, and so on. So more than 2,000 genes have more than have 3 transcript variants, and so on. So about 1,000 genes will have more than 10 variants. So some genes has many different variant transcription. So for example, like in this gene, the gene is the FGFR2 genes. This gene was encoding for a cytokine that played a role in the embryonic development. So it's a carrier having a mutation in these genes. It can lead to the abnormal in the development of the skull and the skeleton during embryonic development, and led to the consequences that the carrier could be intellectual disability when he or she was born. But anyway, you can see that this gene is another really big genes, and including many different exon here. So the first exon in here is the 3' ETR actually. In here we have different transcript variant of the genes. So for example, we compare between these two one. So the exon here is including in this variant, but somehow it's lacking in the second variant. Meanwhile, this 2-1 here will exist in this variant, but somehow lack in the other splicing variant. So let's watch a small clip demonstration about RNA splicing. I don't know, remember if I have shared sound.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay, so now we will have a small break, a 5-minute break before we come to another part, translation. Okay, 5-minute break. Okay, so I received a question during the break. Another time, just remind you, if you want to send me a question, please send it us to everyone, because the other student might interesting in the problem too. Okay. So the questions is related to the transcription process. Wait a minute. So how does the cell decide which mechanism, on which mechanism it decides which exon to skip? So this is a very interesting question. Till nowadays, scientists still working on that, and they have provided several different mechanisms. But one of the mechanisms that have been agreed recently, been proved by experiment, is when the splicing sum come down and trying to remove the intron. So some intron will have a variant or mutation which is in the location nearby of the which is the region nearby to the exon. So this is intron, and this is the region located in the intron but located nearby the exon in here. So the variant, the DNA sequence in the intron in here, might have an effect on the splicing sum and decide of those which exon to putting in the mature mRNA, and which exon will be skipped during splicing process. Okay, and there is more variants. There are more mechanisms that you can find on this paper. Okay, so I will put the link in here to you. So this is a very recent research published on 2015. So this is a very interesting research published recently in 2015 on Biomed report. We study about the mechanism of alternative splicing, just released like 7 years ago, and they have provided several hypotheses how the splicing process was put in. So the three main possible mechanisms: exon shuffling, exonization, and consistently splicing exon. And all the information about research they have provided in the paper here. Okay. All right. So come back to our lesson. Any other question? All right. So three main components that played a role in the translations. First is the mRNA, which is used as a template. So this is the template. For the translation. So the ribosomes, which is composed by two units, the last one and the small one, is play a role as a machine to synthesize the polypeptide. And the last one, tRNA here, is play a role as an ingredient. So the tRNA here is have a safe of the pre-leaf grass, and in one direction is have an anticodon is the pre-nucleotide that binding into the template here, and in one end it's attached to amino acid. The ribosome, which play role as machine to synthesize the polypeptide here, have two subunits. We call it a large subunit and a small subunit. But a very, very recent publication published right in 2019 say that small big in Arabidopsis mitochondrial ribosome. That means in Arabidopsis, a plant that was used as a model for many genetic research in plant, the small subunits even bigger than the large subunit. So we call it the small subunit, but it had the size, the 3D structure size, even bigger than the large one. Okay. So in some species it's like that. So the tRNA here is have a safe three-leaf grass, and in one it's attached to the amino acid, and in here is the anticodon. Okay, which is coding for the tRNA. So this is the universal genetic code that have us to translate from the information of mRNA to the language of polypeptide. So for example, if we have a codon called UAU, so how to deal with it? So we go from the first one here, A, so we come here, and U. So that means it's encoding for tyrosine. Okay. And so, for example, GCA, let's GCA by the same way, come from the first letter here, G, and next one is C, so we go to C, and the last one is A, so is encoding for alanine. So GCA is coding for alanine. Okay. So the translation process start one or a small subunit combined with a large subunit of ribosome and combined to the mRNA as a template. So we have mRNA here. But the translation would not start right at the position of the attached site. It will slide until it reaches the start codon. So AUG here, this is the start codon. So that means the position here to the beginning is the 5' UTR, right? 5' untranslated region. So as soon as it's reached the start codon, so the tRNA carrying the start amino acids, methionine, will come and binding into the sequence. So the tRNA carrying methionine will have the anticodon U, A, and C, which is complementary sequence for AUG, the start codon. So in the ribosome, there's two positions. The first one here, and the second one. Next, the next tRNA will come and binding and release the amino acid here. The catalytic site will enhance a polypeptide link. Polypeptide is actually a covalent link, so it's really, really stable. between methionine, the first amino acid, and the second amino acid one, and then it the methionine form a peptide bond in here, and the tRNA that carry methionine was just out of the position, the first position, and the tRNA here was transferred from the second position to the first position. Yeah. Okay, and another tRNA come and fill in the second position, and so on, so on. We have the tRNA. So the peptide bonding here from amino acid, we will have the alpha carbon, which is the carbon in the middle here, binding to the side group, and we have called it amino acid because it's have an amine group. And the carboxyl group, or we call it the acid group. So the peptide bond will form from the amine group of the second polypeptide, second amino acid, with the first with the carboxyl group of the first amino acid, and form one water and one peptide bond. And as I say, this is a covalent link, so it's really stable. And meanwhile, in DNA, we have the 5' and 3' direction. In amino acid, we have also, okay, wait for me, I open my paint file. Open a new one. So, excuse me, can you turn off your microphone? Can you mute yourself, please? Thank you. So, meanwhile, in DNA, we have the 5' direction and 3' direction in DNA. Then in the polypeptide, we have also the amine group, or the N terminal, and the C terminal, carbon terminal. Of the amino acid sequence. Okay. And the translation process finished, and since the ribosome reached the stop codon. So we have 3 stop codon: is UAG, UGA, and UAA. Since it reached the stop codon, it's when release the polypeptide and finish the translation here. And the polypeptide will directly go to the post translation modification to form the 3D structure of protein. So let's watch a small. clip to demonstrate how translation process. So this is the nucleus, and this is nucleus membrane, and you can see here the yellow region in here is the hole in the nucleus membrane. So we have the mRNA, the mature mRNA here have been finished the maturing process inside the nucleus membrane, and it go into the cytoplasm through the hole in the nucleus membrane. Okay, to start the translation. Oh. So we have ribosome, the small subunit, and the large subunit come and binding into the mRNA to start the translation. So we have the floating in here is the tRNA. The tRNA carrying the suitable amino acid. And we have the product is a polypeptide, which is marked in the red string in here is a polypeptide. So you can see here each tRNA come and give the ribosome will make a peptide bond between the amino acid of the polypeptide chain here with a new amino acid that the tRNA bring along. Okay. So we see that a tRNA here, which is have a red dot in here, is this red dot is the amino acid that the tRNA carry to the ribosome position. Synthesize the protein polypeptide chains. So different type of amino acid. Oh. Different type of amino acids. So the polypeptide here. So as soon as the polypeptide is being synthesized, it start to through post-translation modification to form a 3D shape of the protein. So as soon as the protein as soon as the polypeptide was being synthesized, it had been through the post-translation modification to form its higher structure. So the process called protein folding. So there are four main levels of the structure of protein. So the first level, the primary level, is the sequence of the amino acids. So here, this is the first degree of proteins, first level of protein structure. So the second one is a loop here. For example, the formed by the hydrogen bond from one amino acid to the other, the hydrogen bond, we make to form the alpha helix, which is structure helix here, or sometimes it calls from another structure called the beta helix. Sorry, beta sheet. It's like a sheet of paper from here. So this structure, the secondary structure, the alpha helix or beta sheet, will combine together by several bonds that not covalent bond. This covalent bond could be the hydrogen bond or ionic bond, disulfide bond, one-way bond, or hydrophobic bond, to form a third structure here with several structure of alpha helix and sometimes beta sheet to form a third structure. And in some protein, in several protein, some subunit even combine together to form a more complex structure called the quaternary structure protein. For example, in the hematology here, we have three ternary structure of hematology: 1, 2, 3, 4. We have 4 subunits of hematology combined together to form a hemoglobin with the HAM. That means the iron atom inside. So let's watch a small demonstration about the structure, different structure of protein, and how protein was maintained its function due on the structure of it.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas on the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Databank.
So this is a very interesting data bank. Beside of the general data bank is the protein data bank. So if you go into protein data bank, you type the 3 letters coding here, it will lead into the 3D structure of the protein here. For example, I type 3W14 in the search panel of the protein data bank. It will provide me the 3D structure of insulin. So let's take, for example, I'm going to the protein data bank. Here, RCSB page. And I type D314 Y14. So insulin receptor. So this is the 3D structure. We form a structure of we can see it in 3D structure here. Click on this, wait a few seconds. It's really slow because it contains a huge amount of data. So wait a few seconds more. In here you can export. So here is the label ribbon of. You can pin it in the 3D structure to explore it, to investigate if how it was happening. So I even can zoom in different type of it. Okay. Anyway, it's a very interesting tool. Okay, so the different structure level in here. So the primary structure is formed by the peptide link, which is a covalent link, and the covalent link is really stable to temperature. And but the higher structure, the secondary structure, tertiary, is formed on the non-covalent link. That's why it's very sensitive with other factors that like temperature. Okay, so it's easy to use temperature to break the 3D structure of protein, because all the higher secondary and higher structure of protein was formed by the non-covalent link, like hydrogen bond or one bonds or hydrophobic bonds. So as a consequences from one sequence of amino acids can based on the different post-translation modification can form different structure of protein. So with one polypeptide sequence, we will have different isoform of protein, and different isoform will maintain different function. So as a result, from one DNA through a splicing process, will form different type of RNA. So we have different RNA variant of one DNA, one genes, and different RNA variant through the post-translation modification can form different type of protein. So as a result, the number of proteins will be much higher than the number of genes, always greater than the number of genes. Okay. So now to finish today lecture, I give you a exercise. So this is provide sequence of a genes with the full sequence that had categorized for you. So this is the promoter, this exon, intron, exon, intron, and exon. So it's a gene with 3 exon and 2 intron. Please identify for me where does the transcription start. What is the sequence of the mRNA precursor? That means the immature mRNA. Okay, sorry, immature mRNA. Follow the English pronunciation. The mature messenger RNA. Where does translation start, and what is the peptide sequence? Write down for me the first 10 residues of a peptide sequence, and where does the translation start? Okay, please help me to identify and answer the question.
So is this the coding strand? Is this sequence the coding strand or the peptide strand?
So this is the coding strand.
Okay.
Yeah, so this coding strand from the 5' to 3' of the coding strand. Sorry. So for your convenience, I will copy the sequence here and put it in the chat box for you. Okay. Okay, so I have the first answer. Wait for me a minute. And
Excuse me, can you take back the file? Like, can you go back to the slide?
yeah, here.
There's some wrong here. There's something wrong here. Like the mature messenger RNA is wrong. I will take it back. That the two got replaced.
Okay.
So with you. Yeah, I love Python. Yo.
Okay, so the first question, where does transcription start? So we have identified it, studied in today's lecture, right? Those transcription start. Oh, in here, right after the promoter, right? So the transcription start in the position +1 here. Transcription starting site. Okay. So the transcription starting in the exon 1 in the position here. So this is the position +1, or the transcription starting site. Second question, what is the sequence of the messenger RNA precursor that the immature mRNA? What is the sequence of the mature messenger RNA? So we came back to the same slide. So this is the premature mRNA. So it including also all the exon and all the intron. And the mature mRNA is including only the exon. So as a result, we can see that the precursor is including all the sequence from here to here, and the mature messenger RNA is including only the exon. So exon 1, exon 2, and exon 3. Okay. All right. So your answer is correct. So where does translation start? Translation
starts on AUG or on the AUG.
Yeah, so we come from the transcription starting site, we scan in here until we reach the AUG or ATG on the sequence. So the first ATG.
I found the index is 68 or 69, like depending on if the string is 0 index or 1 index.
I'm sorry, what is the question?
I use Python to do it. Like I found an
okay. ATG. Yeah, but the first ATG, not the second one. Okay. What is the first
one? Yeah, the first one. The first
so in this position, right? Or in other? Okay.
Yeah, position 68 on Python.
68. Okay, great. Okay. So you use Python to use it. So online we have several Python's pipeline to guide us to how to find out the translation starting site of a genes. So you can use it up if you like, no problem. Okay. And what is the peptide sequence? Can you put it in the chat box for me? So we have ATGs coding for methionine. We have the next 3-1 here, next 3-1 here, here, here. So you can use the universal code. So for example, AAT. T is replaced by U. So it's asparagine, okay? ASP. And so on. So on like this, you have 10 residual from it. And what does the translation stop? So this question is tricky. So you have to follow this ATG, 3 nucleotide, 3, 1, 3, until you reach the stop codon, okay? Remember that if you have a stop codon like here, here.
There is a UGC that ends at index
So here. Okay, so we have the stop codon. Okay, so that means that the translation will stop here. So that means all the region here is a 5' UTR, and all this region here and to the exon 3 is a 3' UTR. Okay. All right, so I guess that is the end of today's section. Do you have any question, comment? Okay, so since you have no more question or comment, I will finish today's section here. Thank you very much for your attention and see you on Thursday.
Goodbye, Professor.
Goodbye, Professor.
Goodbye, Professor.
Goodbye.
Goodbye, Professor.